Thanks, Faye. I really appreciate that. Yeah, so this is going to be mostly a talk about my master's research, the University of Georgia. But at the end, I'm going to sort of tie in what we're doing nowadays with Japanese beetles and how it relates to the big picture of monarch conservation and also beneficial insect conservation. So I'm going to start with some background and then talk about the 4 experiments I did during my master's, 2 on larval monarchs and 2 on adult monarchs. And then I'll talk at the end just a little bit about the research we're doing on Japanese beetles. So to get at this question, we want to look at aspects of survival, reproduction, behavior, and whether there are differences between larval and adult reactions to neonicotinoid insecticides, as well as some interactions between chemicals that are found in milkweed. So milkweed is a toxic plant that monarchs have evolved to deal with. So there's some a little bit of interaction there that we're going to look at. So just to get you oriented, neonicotinoids are a pretty popular insecticide that are used in agriculture. This is a bit of a misleading photo. Usually they're not sprayed foliarly. They're generally treated in the soil and plants take them up. Since they're systemic insecticide, plants will incorporate them into their leaf tissues and express them as well through pollen and nectar. And this is an important avenue for how beneficial insects can be exposed to them. And it's good to note here that only about 5% of the active ingredient ends up in the target plant. So the rest, 95% of the active ingredient, goes into the environment and can be spread through the watershed and taken up by plants that were not intended to be treated. So throughout this talk, I'm going to be mentioning clothianidin and imidacloprid. They're just types of neonicotinoids that are the most commonly used in agriculture. So in agriculture, this graph is showing the total load of insecticides over time. And in red, we can see neonicotinoids, and in pink, we can see all other insecticides. So as you can tell, since the late 90s, neonicotinoids have really become the dominant insecticide that's used in agriculture. And this is important for, aspects like we talked about, spreading to non-target plants and those sorts of things. And if we look at the worldwide scope, I know this is a small map. The idea here is that they're used across the world generally. And this map is a little outdated. In Europe, they've since been banned. I think it was like in 2018. So disregard the Europe section of this. But the point here is that they're used across the world pretty heavily. And before I get too deep into the monarch side of things, I wanted to talk a bit about bees. Honeybees and bumblebees have been extremely well studied in their responses to neonicotinoid insecticides. So we've seen reductions in foraging, which leads to some interesting effects on fecundity and reproduction and those sorts of things, as well as some behavioral effects. So we see grooming behavior reduced in bees at really low concentrations of neonicotinoids. We also see their flight ability impaired, as well as their navigation ability. And here I'm citing just 4 papers that talk about these things, but realistically, there can be 100 papers because there's just been so much research done in this area in bees, and it's really lacking in monarchs. So, in contrast, well, in butterflies in general, and in contrast, we wanted to talk about monarchs because they're a really good study system. They're very easy to rear. The caterpillars sort of, feed on plants and they grow. They're also very scientifically important in terms of their yearly migrations, as well as their interactions with chemicals in their host plant species, milkweed. So milkweed has cardinalides, which are toxic chemicals. They are sort of a good deterrent for herbivores, but monarchs have evolved the ability to sequester these toxins and use them in their tissues. So when they are, eaten by predators, the predator knows not to eat them again. And that's why they're the larvae are brightly colored and the butterflies are also brightly colored. And a good example of this is Lincoln Brower's work in the 70s, where he feeds a blue jay a monarch, and you can see the blue jay promptly vomits. And for the rest of that bird's life, it's never going to eat an orange-colored butterfly again. So that's a good example of mimicry and aposematism and those sorts of things. And on the topic of milkweed, there's over 100 species in the US. There's really a lot. And they all differ in their concentration of cardinalides, their polarity, the toxicity. So if you take something like swamp milkweed, which is on the top left, it has next to no cardinalides. And in contrast, tropical milkweed on the bottom left has the most cardinalides. And it's actually so toxic that if you get the latex on your hands and rub your eyes, you risk going blind. It's how toxic it is. And the point is they all differ. And even within species, if you rear them at different latitudes, they can develop different expressions of their toxicity. So we'll get into that later in the talk. And in terms of the migration, monarchs are the furthest migrating species that's been studied to date. They migrate from as far north as Canada all the way down to central Mexico, which is 5,000 kilometers in a year. And that's in the eastern population, but in the western population, they migrate from further into Oregon and Washington down to coastal California. So these two populations are genetically very similar, but their migrations do differ quite a bit. And we've also seen a lot of declines in monarchs in the recent years. So you may have seen something similar to this graph. In the Western population in 2021, there were only 1,600 individuals counted for Thanksgiving and only 1,000 at New Year's in the Western population. So they really took a deep dive there. And that line on this graph is just showing that the actual participation counts efforts has gone up while the numbers have gone down. And then in the Eastern population, we see a similar decline, not as drastic, but it definitely did bottom out around 2013 and 2014. So generally, the point here is that monarchs are declining, and there's a few explanations as to why. One of the explanations is habitat loss. So monarchs need nectaring plants in addition to milkweed for reproduction. Adult monarchs have to interact with a lot of different flowers and drink nectar. And it's not just milkweed that they interact with, but also on top of that, the numbers of milkweed have been declining as well. And this is because of agriculture and because of urbanization, habitat reduction, the use of herbicides, the list goes on. And insecticides are also suggested to correlate with their declines. And that's what we're getting at today. So it's important to talk about how much of neonicotinoids monarchs can actually come into contact with in the wild. If we look at their responses to the toxicity and those sorts of things, it's interesting, but if you don't have numbers from the wild, you don't know how to gauge that with what they're actually coming in contact with. So a couple of studies have done milkweed in the borders of agricultural plots, and one of them found up to 24 nanogram per gram, which is equal to parts per billion. So it's quite a small concentration in the grand scheme of things. And another found 56.5 nanogram per gram. And these are all bordering agricultural fields, drift and accidental exposure and those sorts of things. But if we look at greenhouse reared, there is a large potential for exposure to higher concentrations of neonicotinoids. So this study treated potted plants, potted milkweeds, with spraying and drenching neonicotinoids. And they actually ended up with quite a lot after 2 and 4 weeks. So here we're on the log scale, so that 3 concentration would be around 1,000 parts per billion, which is quite a bit in comparison to the 24 and 56 that we're looking at in the wild. So this, greenhouse reared milkweed that you buy at Home Depot and Lowe's and those sorts of things, if they're treated beforehand, they could potentially have a whole lot of insecticides in them that you're putting out in your yard. So if you missed Niranjana Krishnan's talk in the department a couple months ago, it was really good, and she got into some of these questions about how monarchs are interacting with neonicotinoids in terms of their the methodology and the mechanism for them. So she found that there was a large amount of arrestic diesis, so the pupation ability of monarchs is interrupted by neonicotinoids, and that picture there is an example of that. That's actually from our work that I'm about to talk about, where we saw the same effect. So essentially, when monarchs are late, the larvae are like late instar, so around fifth instar, like this one is. If they're sprayed in their cuticle with even low concentrations of neonicotinoids, they can actually be interrupted in their pupation ability. So that part of the hormone that is released is interrupted because of neonicotinoids. And if you spray the same amount to a young larvae, it's actually able to clear it before it pupates, and it'll be fine. So the timing of when a monarch is exposed to neonicotinoids is really, really important. And this will change the actual LC50 that we see based on the methods that we use. So I just put up some numbers from her work. So she found 130 nanogram per gram and 74 nanogram per gram LC50s on larvae. And keeping in mind that 56.5 that we saw in the wild, it's a little bit above that, but it would still, that number could kill some monarchs. Okay. So now I'm going to get into the work that we did. And the goal was to quantify the sublethal and lethal effects of neonicotinoids at 2 life stages. So here's a basic life cycle. We have our egg, larvae, and pupae and adult. And here we're going to start with the larval work. So 2 experiments here. And those experiments were, the first one was low dose neonicotinoids and looking at their survival, growth, and flight performance. And then we followed that up with higher doses where we were looking at their survival, again, their growth. This time we looked at their strength and we compared effects between 3 different milkweed species. So I wanted to mention, and I guess I can mention it here, that there was an early study that looked at these ideas on larval effects of neonicotinoids, and they actually found that really low doses, like 15 parts per billion, would kill larvae. And that's sort of what informed the first study here. So more recent work has showed that it takes a lot more. But this first one that came out by Lundgren and Pasinka in 2015 showed some really low concentration effects. And that's what our first study was sort of based on. So we use both clothianidin and imidacloprid. And again, this is really low concentration. So these were the realized concentrations. So we sprayed 5 and 15 parts per billion or nanogram per gram. And this is what was the residue that was recovered at the end of the experiment. For this experiment, we only used swamp milkweed. We treated leaves every day for 5 days. And then at the end, we measured their survival. We weighed pupae, calculated their growth rates, weighed the adults. We scanned their wings on just a flat scanner, and we're able to calculate their wing area. And then we also measured their flight performance. So once the treated larvae eclosed as adults, we glued their thoraxes with fishing wire and attached them to this flight mill. And there's a ball bearing in the middle that lets it, near frictionless spin around. And every time that flag passes through the infrared beam, it sends a signal to the computer and gives us a time that it took to go around. So we can calculate the distance that it flew, as well as the speed that it took. And then we followed that up with higher doses and 3 milkweed species. So we again looked at clothianidin and imidacloprid, but this time our recovered concentrations were between 4 and 61 nanogram per gram. And we treated them, we treated the plants as a whole this time, and we enclosed them in tubes so that once the plants were treated and once the larvae were on the plants, they could eat and they wouldn't have to be disturbed. So we sort of reduced some of that stress that we were introducing in the previous experiment. And we looked at effects of swamp, common, and tropical milkweed, which constitute low, medium, and high cardinolide concentrations. So in this experiment, we again measured their survival. We weighed the pupae, calculated growth rate. This time we had obvious deformities. So we had, as well as aristidicdiasis, we also showed deformed pupae. So even though they were able to pupate, some of them were actually like discolored and deformed as pupae, even though they still made it through the cycle. So we quantified that. And we also measured their wing area and their grip strength this time. So this used a similar mechanism as the flight mill, a similar computer system. But this time it's measuring our newtons of force. So we would just let them grab onto that rod and pull them up at a constant rate, and that will tell us how much they exerted before releasing. So in our low dose experiment, we saw no effects. Spoiler alert. Here we're showing growth rate on the y-axis, and on the x-axis we have a control. We have our metacloprid 5 and 15 parts per billion sprayed, and clothianidin 5 and 15. And we're showing means and standard error, as well as the individuals jittered on this plot. So the story for this experiment is that there were no effects at these low concentrations. We saw no mortality. We saw no development effects, no flight effects throughout this experiment. And I'm at the end. Great. Sorry. Where were we? Okay. Which is important because it contradicts that first study that said low concentrations are lethal. Here we're showing that's not the case. And not only is it not lethal, it does nothing at all. But this is consistent with more recent studies. So in that follow-up experiment, we saw, so here on this graph, we're looking at proportion to form. We have our control in blue, imidacloprid in green, clothianidin in orange, and we have our low and high concentrations for each of those types of neonicotinoids. And then we're colored by our milkweed species. So there's a lot going on in this graph, I know. But so we have swamp in blue, common in green, and tropical in black triangles. And we saw that common and swamp milkweed that were treated with clothianidin at the highest concentration had more deformed individuals. And those were the only two groups where we saw significant deformity. So swamp had the highest, and then common milkweed had an intermediate effect of deformity. What's interesting is swamp was our lower cardinolide, common was our intermediate cardinolide, and tropical was our high. So we sort of saw an inverse gradient in the effects of neonicotinoids here. And then for the number that actually enclosed properly, only swamp had a reduced number of enclosed individuals treated with clothianidin at 61 parts per billion nanogram per gram. We also saw swamp has significantly lower wing area, so they were smaller, treated with clothianidin and at 61 nanogram per gram. And then that same group had significantly lower grip strength. So the swamp milkweed here, treated with clothianidin at 61 nanogram per gram, was deformed, enclosed less, had a smaller wing area, and had lower grip strength. So to summarize, the low doses, we saw no effects of anything, survival, size, flight performance. At higher doses, we did see effects of deformities, death, size, and strength, but it depends on what species of milkweed monarchs are viewed on. Okay, so next we're going to talk about adult exposure, brief intermission. So there's been a lot less work in adult monarchs than there has been in larval monarchs. And we see 3 studies in the literature to talk about this. So in 2015, Vera's work showed that there was no significant reduction at 30 nanogram per gram of imidacloprid. And then in contrast, in 2019, David James found that 25 nanogram per gram of clothianidin did significantly reduce lifespan on adults. But the more recent study by Niranjana Krishnan found no effects up to really high concentrations. So the 2 experiments we're going to be talking through here are low to moderate doses, looking at monarch survival, reproduction, and behavior, and then following that up with a high dose effect and looking at survival, flight performance, and reaction time. So in our first study, we used a wide range of concentrations. This time we don't have to worry about how much is in leaves and that sort of thing because we're just treating nectar. So we mixed between 0 and 500 nanogram per milliliter into 20% honey water. And we fed monarchs every other day for 10 days. So they were fed 5 times total. And we weighed them before and after each feeding, so we knew how much each individual actually consumed. Of course, we recorded their survival. We looked at their mating twice per day. We recorded their activity levels. So we had them all in these cages here. And we would look at them for 10 minutes each time. And in every cage, we record whether monarchs were feeding, mating, contacting other monarchs, or if they were just sitting on the walls of the cages. And then from those, we took a subset of the mated females and we let them lay eggs on milkweed plants. We counted the eggs they laid, and then we recorded how many of those eggs successfully hatched. And then with everything else that wasn't in oviposition cages, we just let them sit in the fridge without feeding them and recorded a longevity for them. And then in the follow-up study, we looked at 0, 900, and 4,000 nanogram per mil of just clothianidin. And we measured their survival. We flew them again, as we did previously. And then this time we conducted a drop test where we took each monarch at arm's length and had a stopwatch and let them go. And once they opened their wings, we would stop the stopwatch. So it was interesting because some of them would open their wings immediately, as you might expect. Others would let themselves hit the ground before opening their wings. So results for this, at moderate doses, here we're showing change in weight. There was no significant effects. So here on our x-axis, we have the dose that each monarch has. And you can see each monarch jittered there. So they're not perfect bins because we knew how much each monarch actually consumed. And then we just had an overall line. But we did see significantly reduced number of matings that males had. At higher concentrations, males mated less often. And then we also saw that the mated females laid less eggs at higher concentrations of imidacloprid. And then in the follow-up study, we showed a reduction in survival in both treatment groups. So both at 900 and 4,000 nanogram per milliliter, they had about 60% survival. And then in the highest concentration, they consumed significantly less nectar. And at both of the treated concentrations, they responded much slower. So that's a difference of 0.3 in the controls reacting immediately, and then in the treatment groups up to a second reaction time, which is quite slow. They were the ones that hit the ground. And then in the flight variables, we saw that both treatment groups had reduced distance flown and reduced speed in the highest concentration. So to summarize, we saw no effects below 500 nanogram per gram, but at 500 nanogram per milliliter, we saw effects on reproduction. So that's where the males mated less often and the females laid less eggs with imidacloprid. And then at higher concentrations, we saw effects on survival, flight performance, and reaction time at these higher concentrations. So to pull it all together, basically we see that field relevant doses could have negative effects on larvae. So at that 56.5 number that we saw in the wild, that's pretty close to our 61 nanogram per gram that we treated with and saw some negative effects depending on the milkweed species. But that is sort of dependent on the timing that monarchs are treated with neonicotinoids, as well as the host species that we mirrored on. And we see that it takes a lot higher concentrations for adults to have negative effects. And when you combine that with the fact that the concentration in nectar is much lower than the concentration in leaves in the wild, the potential for negative effects on adults is much more unlikely than it is on larvae. So to pull it all into the next steps, what does it have to do with Japanese beetles? Well, Japanese beetles are the recipient of most of the insecticides that are sprayed in Minnesota. So it's important to think about, how we're treating foreign invasive species and how that's going to affect beneficial insects, right? So what we did over the summer on Japanese beetles was we looked at some biorational insecticides. So we looked at BTG, which is Galleria, which is more specific to Japanese beetles. And we looked at Bavaria bassiana as potential treatments to treat Japanese beetles with instead of using conventional insecticides. And we saw that BT was pretty promising, very promising actually in the product that we use, Beetle Gone. It killed 100% of the beetles at label rate in 96-hour bioassays. And then Microtrol was sort of effective. It killed about 50% in 96-hour bioassays. But the question is, are these better than conventional insecticides? We don't know. No one's used these biorational insecticides on monarchs or on bees to see if they are as host specific as they say they are, or if there's something else in these products that could have effects on beneficial insects. So we need to test these insecticides on monarchs and other butterflies to see if there is potential for negative effects and those sorts of things. If we're actually saving time and saving resources by switching to these instead of conventional insecticides. Okay, that's all I have. A lot of people to thank. My advisor at University of Georgia, Sonia Altizer, was instrumental in a lot of this work. I had a lot of help. Vera was extremely helpful in all of this work, and she hired me, so it's yeah, thanks.
Any questions for Cody? What's your mechanistic hypothesis for those differences among those different plants that you saw? Like, are they just less suitable hosts? Because you have variation in your control amongst those different plants in the lot of sense that you're measuring.
Yeah, absolutely. So the short answer is we don't know, because we didn't get deep enough into it. So the hypotheses we sort of had were it could be interacting with the cardinalides, the inside of the caterpillar's gut, or like the leaves on those plants are different. Like they hold water differently. They interact differently. Some of them are thicker than others. So it's sort of like even if you spray the same amount on swamp versus common milkweed, like how much is actually, yeah, how much is actually in there is going to be different.
Is there other differences in what the butterflies or the larvae prefer in nature? Are you more or less likely to find them on some of the plants?
Yeah. So, I mean, I feel like you would be more likely to find them on common milkweed just because there's more of it. And the leaves are nice and big so that they, there's more to eat for the larvae. So I would say they're more likely to be on common milkweed. Yeah.
There's a lot of milkweed as a species.
Mm-hmm.
That's not so it's not really common in nature, but all like and so they quality effects online and that is very close to program inclusive. But nobody else has looked at the synergies of the neonicotinoids and that needs to be perceived.
Thank you.
So I'm really curious about your adult assays and adult exposures.
Yeah.
And I think you showed that when you're higher concentrations of imidacloprid, you saw reduced consumption. Is that right?
Yeah. So.
And so you've got your neonic in some sort of nectar bottle and you're just consuming less. Is that fundamentally right?
Yeah. So they're all force fed. So here's a picture of them being fed. So they're all force fed on these trays. We probe their proboscis and they have to drink. And those that were treated higher concentrations, we saw that over the 10 days that we were feeding them, they just would you would pull their proboscis out and they would put it back in and they were just lethargic and just not feeling to consume that.
So I'm just kind of curious though, did you ever see what happens to monarch behavior if you just starve or dehydrate them?
Not in this study. No.
So I'm just curious how you tease apart some of those toxic effects from some of these other behavioral, physiological effects.
Sure. But I mean, if it's the toxic effect leading to a behavioral effect, then that's still interesting, I think.
Oh, it is, but you just want to be careful about what you're saying is the cause.
Sure. Sure.
Yeah, I have a related question with this. You're measuring weight before and after.
Yeah.
But that could be excretion, water loss, any number of things that you're looking for a change in, and not necessarily how much they consume, right?
Well, it's immediate before and after. So
what sort of time frame?
Weigh them, feed them, weigh them.
Like hours, days?
Like literally weigh them and then take them off the scale, feed them, put them back on the scale. I don't remember. It was 10 minutes each.
Okay, so pretty quick. Okay.
Yeah.
I guess that's a little, for the 10 days. Makes more sense.
Oh, I see. Yeah. Yep. Yeah, that's a good point. Yeah.
nobody's ever looked at.
Mhm.
So back to the adult effects, so whatever effects you did see did not occur until you get to 500.
That's correct.
Well,
Yep, yep. So those, I'll show you those graphs again. So it's an overall effect, right? But really it's driven by that higher concentration here. So if you see the individuals there, they're sort of driving those effects.
So basically, this Lundgren, Jonathan Lundgren, that was master's here. That's the Lundgren we're talking about. Parts of the billion. We're now talking, and then the results and that 25 parts per billion. Now we're at the 600. Without the parts per billion. So this study was, there's three studies that come out at the same time. And they really have changed people's understanding of the effects of neonics on butterflies, and it's much less than bees. No comparison to the effects on bees.
Yep.
And Mira and Donna was somebody the two of us met in this process. And one of the things we figured out is these toxicology studies like Lundgren did are 4 or 5 days long, and you get completely different data than if you do a chronic study. So most of our clinical studies are chronic. So if you actually grow something, even if it's his larvae weren't dosed every day, they're feeding on the same, the plant was dosed once. Still, a chronic is 1,000 times less lower LC50 in a chronic than in acute. And so most people look at acute and take that data and bring it up to the population level and say, oh yeah, pesticides can huge effect on insects, but what Nara and John found out, before Nara found out in this other study that I submitted found out is no. It's completely biased based on if it's a 4-day LC50 or a chronic study.
Which is a promising result, which is, it's not as like scary as the first few papers that came out, I think. Do you have a question? Thanks.
Work you done here, and then talk about Japanese beetles and looking at those. Have plans also them. You study the best of those is that the next?
Yeah, we hope to do that this year, next year. Yeah, so that would be an interesting part of it. Yeah, I think so.
Systemic, right?
They're not systemic, so they would be sprayed on the plant. But even in this study, like, we didn't treat the soil, we just sprayed the neonics on the plant. So
Are those like, would those be sprayed in greenhouse as well? Is that like
for those biorationals, yeah, I think they're like spot treatment more so. So just like if you see Japanese beetles, you would spray them with those biorationals. Yep. Yeah.
Can you clarify? But you said there were impacts on patient, and you have the text ecdysis.
Right.
Just the facial ecdysis, the pupil ecdysis, or is it?
It is, yeah. So it's pupil ecdysis. Yeah. So like with molting, like there was no issue.
Yeah.
Yep.
Is there a reason that you didn't use, you had your 3 types of milkweed, you didn't, I saw you didn't use Asclepias tuberosa, and just in my experience, it seems like both in the wild and in retail places and in people's yards, it's a lot more common than some of the other ones. It might just not have fallen into that nice gradient, like where you're trying to look at the levels and the plants. I just was wondering if I would use that.
Yeah, so that gradient was what we were going for. So we wanted just one intermediate, and then swamp milkweed has really like hardly any, and then tropical has a ton. So it just so happened that common falls in the middle. But I mean, we could have used tuberosa instead of common as the middle one.
Also.
Yeah.
So it's a technique that is real hard to go to this.
That too.
And you can play. People use this which is determinate, so you can only do the multi-study with it. And then this travel goes into time, so you can do 6 months of research. So it's more like techniques. They would have been done at Cornell, and he's looked at, he has a table of what cardinals and different species. So that information is, I wrote these, and that information.
Yep.
So, Cody, I understood that in Utah, there was a medical code on adult devices, i.e. and that they had a problem with their wings.
Yeah.
So you don't see any problem with wings or wing infections with the monarch?
So, and some of those in that one group that we saw the negative effects on, we saw like pigment issues. So the blacks weren't black. It was like really sort of clear white wings towards the tips. And then we saw like the pupil discoloration a little bit.
That's because that is an effect of versicon, which is released with the basically the peptide which induces the excitement behavior. And so versicon not only does sclerization, but it does, the tanning, pigment.
And the wing inflation.
Yeah, so yeah.
Okay. Yeah. That is really cool.
This fall, she has a recent paper. She looked at 3 or 4 species and the effects of neonicotinoids on ecdysis. And it was different for the different species, but in our lab too, the monarchs, they don't get that far. They have corrupted pupae in some way. Their wings, they don't go on their wings, that's some of it for that.
Yeah. And we in this study, we really just recorded that as failed to pupate or something like that. We didn't really, quantify that sort of thing.
But some species who become functional, they're moving adults, they're probably not going to be able to be functional, and they have purple wings, but monarchs don't.
What species was the other?
We did fall webworm and
Okay.
Val, did you have a question?
Based on your work and the recent literature, how do you think pesticides stack up with some of the other stressors out there for monarchs and habitat loss or herbicides using?
Yeah. My guess would be that these effects, at least especially on adults, are really not much in comparison to other, bigger impacts like loss of habitat and those sorts of things. But that is to say, like, all these things sort of come together. So it's hard to say like this is better than that is better than that because they all contribute, right? Like, if you have a diseased monarch that has nothing else going on with it, it may be fine and make it to Mexico. But if you have a diseased monarch that's been treated with neonicotinoids that has been exposed to herbicides, whatever, it wouldn't, right? So I think it's sort of a more of a everything together issue.
So we also did the field residues, that's the question, in soy, corn, and you're happy to know that there's not much drugs in soybean and corn, but potatoes have some vitamins and D2 and DBT, when you comes up in the residue studies. So more of
Hmm.
like corn and soybeans had much good. So it was all below the food and compost.
Thank you all for being here. Yes. One more.
Unrelated to your
Okay.
talk, but I want to question.
Okay.
Thank you, man. I'm curious if you can get it. I get this question every now and then. And so the conservation of monarchs, people are rearing them and then releasing them.
Mm.
And so there's some concern that if you do that, they don't go all the way to California, Mexico.
Mm.
And so should people not be doing that, or so how do you answer that?
Yeah, I mean, it's a hard question to answer. People have shown that like the orientation ability of reared monarchs is reduced. So they're less able to find the right direction they're supposed to be going. And there's some other like they're just weaker when they're reared versus wild and those sorts of things. But you are at the very least like bolstering the reproductive population, right? So even if they can't migrate, maybe they can reproduce in the summer and contribute to more wild individuals. So I don't think it's a bad thing, but it's tricky because everyone does different stuff when they rear butterflies. So
there's two listers out there, one from here from Minnesota and one from Kansas State. And so this question comes up on the all the time. And they have found that individuals end up in it. It's rare, but they have found them.
Okay, is that it? Thank you all.